Welcome, ladies and gentlemen, to yet another special Berlin Science Week event, this time in cooperation with the Berlin University Alliance and the European Research Council. It's about the transformative effect of science. Quite a promise, and sometimes even a threat. A promise. Over the course of history, and especially within the last 200 years, science has brought enormous improvements to the way we live. Even more so and more rapidly in the last 50 years or so. We use technologies people were not able to imagine a few decades ago. We are in the middle of an energy revolution. We are able to fight formerly deadly diseases. And we managed to survive with 8 billion people on this planet, a planet that has been inhabited by a few hundred million for almost all of history. A danger. Also, because of our progress, the Earth is sick, as we all know. We are on the verge of ecological, political, and societal breakdowns. Because we are a global society, new diseases can spread quickly, as we have experienced, and we are not able to control them in time. Sometimes it seems that for one solution science delivers, there is at least a new challenge, at least one new challenge. Is it that what we mean when we talk about the transformative effect of science? Or are we just getting started? Will science lead us into a new, more sustainable future? So let's talk about science. Let's talk about the places where it happens, the universities. And a network is a promise too about the Berlin Science University Alliance, the Berlin University Alliance. Tonight, we are going to learn more about the research being done by the BUA universities and how it is being stimulated by the alliances as well as by the European Research Council. My name is Jan-Martin Viada. I'm a journalist for education and research. And as your moderator tonight, I'm happy to give the floor now to Professor Dr. Sabine Kunst. She's the president of the Humboldt University and the speaker of the Berlin University Alliance. Sabine Kunst, great to have you here.
Thank you, Jan-Martin Viada, for the kind introduction. Dear President Lept, dear ERC grantees, dear colleagues, dear friends of the University Alliance, ladies and gentlemen. I have the privilege to bid you welcome here in this fantastic location in the name of the Berlin University Alliance to the joint event of the European Research Council. We are very proud that you, Mrs. Lept, are with us here today as your first visit to Berlin in your new role as President of the European Research Council. Professor Lept has started her new role only a couple of days ago on the 1st of November. And I'm very curious about the talk she will provide us. The ERC is one of the most prestigious scientific funding institutions worldwide. It provides funding for ideas created by scientists in a bottom-up approach, funding for basic, fundamental frontier research. Receiving a grant by the ERC is certainly one of the crown jewels of scientists. The title of the session today is The Transformative Effect of Science. Why should we discuss about this? As researchers, we want also to show how our scientific effort contributes to effectively transforming our societies. For universities, this effort can be labeled the third mission, going beyond research and teaching. Only a few developments have shown drastically the value of research than the COVID pandemic and the rapid evolution of vaccines with the help of the ERC, as we all know. However, if we consider the developments with regard to the research funding, Europe still lags behind other continents. Fundamental research is sometimes under pressure by policymakers. The expectation exists science should help them solve the difficulties and pressing needs, the great challenges of today. And indeed, this is the main tenet of the Berlin University Alliance: contribute to the solution of complex global problems and the search for interdisciplinary answers. To showcase the value of frontier research, we need to become much better at explaining the complex relationship between science and society. Fundamental research is often so specific that even other academics have difficulties in understanding the essence, and the results may seem irrelevant at this time of their finding. But in the course of time, they may become revolutionary. All science effectively relies on the insight provided by prior fundamental science. Universities and public research institutions perform more than 3/4 of total basic research. And to ensure continued support for basic research, we need to make sure that it is understood that fundamental science serves to learn to understand how things work and to push the boundaries of human knowledge further. It is necessary to provide fundamental researchers with the trust, time and resources to allow them to contribute their dedication and turn ideas into scientific breakthroughs. The funding system of the ERC is one example of doing this. The Berlin University Alliance is a vibrant partnership, and each of the 4 institutions is proud to have a number of ERC grantees. 4 of them will provide you with a brief insight into the essence of their projects and give you very distinct examples of the transformative potential of fundamental science. Professor Rillich, Professor Schneider, Professor Begüm, and Professor Södeker. But before they will have the word, I'm very much looking forward to hear your words, Mrs. Leptin, about the role of science and the European Research Council. I'll hand the micro back to Jan-Martin Viada, who will briefly introduce the next steps of the event today.
Thank you very much, Sabine Kunst. And now it's a huge honor for me to introduce Professor Dr. Maria Leptin to you. This, as Sabine Kunst has just pointed out, is one of her first official appearances as the new president of the European Research Council, since she has taken office just 5 days ago. Prior to that, she served as director of the European Molecular Biology Organization, bringing together 1,800 leading researchers. She also established a research group in Heidelberg at the EMBL. The ERC, as you all know, has proven to be the beacon of excellence of European research policy. Professor Leptin will speak now about how science is related to technology, innovation, practical application and growth, and why science is so central for addressing the many societal challenges that lie ahead, and also so central for creating a sustainable future. Maria Leptin, everybody.
Können wir das wegnehmen? Thank you. Liebe Kollegin Kunst, dear colleagues and friends in the audience, dear ERC grantees, the most exciting part of this evening, ladies and gentlemen, I'm delighted to be here as part of this Berlin Week. It's a great opportunity to celebrate science, and I'm glad that we could put this event together with the Berlin University Alliance. I specifically want to thank my predecessor, Jean-Pierre Bourguignon, who's here in the audience, and Professor Günter Ziegler from the Freie Universität for having mediated this. And of course, Johannes Vogel for allowing us to be in this fantastic space here. And indeed, I'm proud that this is my first event as president of the European Research Council. I've spent 3 days in Brussels. The first 2 days of the week were holidays. So, for those of you who don't know me, I'm a development biologist and immunologist. My career has taken me throughout Europe and the US, and now to Brussels. One of my aims will be to build greater appreciation of what the ERC does and to foster a more positive attitude towards research in all fields among all citizens in Europe. And one of the approaches of that will, of course, be communicating better the success of ERC grantees. The ultimate proof of what the ERC does, after all, is the work of our grantees. And so I'm really delighted to have 4 of them here and look forward to listening to them. Jean-Pierre, my predecessor, realized and also made it clear to me that the best ambassadors for the ERC are in fact the brilliant researchers that we support. So, like I said, citizens in all countries in Europe should know what their ERC grantees are doing, what they're finding out, and should take great pride in them. But I'm here today to talk about science in general and why society should support it. Science is, at its core, the attempt to understand the world around us and how that world functions. I, for one, believe that this alone is a worthwhile end, and that's what will be my theme throughout my tenure. I understand it may sometimes be difficult to persuade governments and citizens to support us at the levels that they do, and that I believe are minimally necessary, if understanding was the only result of research. Let me also be clear that when I say us or we, I don't mean only the natural sciences, but all academic research. Even though I will focus today on the natural science, I want to make it clear that I consider curiosity-driven research in all other fields too: philosophy, archaeology, economics, psychology, so humanities, social sciences, everything. Important and of interest for all citizens and their governments. Governments do, of course, support arts and research into humanities to some extent. In Germany, we're very lucky, we have good funding, but nothing like the level that they support the natural sciences. And that's what today's topic is about. So let's ask, what do people actually want from the natural sciences? I think mainly if they're asked what output they want improvements in their standard of living. And that's largely based on material of wealth. Now, economists have, of course, been studying the factors that create wealth, the factors of production, land, labor, capital, for over 200 years. Only in the last century did it become clear that not all of the historical growth in production and wealth can be explained through existing knowledge, but a large component requires a new factor in production, namely technological progress. Imagine, for instance, a farmer that can grow enough food to feed him and his family. I say him and his family because it was only men at the time. And then they can produce enough food for themselves. Add another farmer, they can produce twice as much, but per farmer it remains the same. But if you give them capital, for instance a plow, then they become more efficient, more effective, and grow more. Give them 2 plows, now they can both do more. But eventually, give them more plows and their children can help. But eventually you will saturate the world with plows and you cannot make further progress. And they then have to look for something better, something new to continue to improve. Better tractor, or perhaps fertilizers, or genetically engineered plants. So real growth per capita in wealth ultimately comes from new technology, and that's what the citizen is interested in. But where does that technology come from? Ultimately, all technologies use natural phenomena. We work with nature, they're based on nature. We can't just invite anything we can dream of. That's what movie makers and science fiction writers do, but we know that that can't work. And there have been some hilarious predictions from those that just couldn't have worked in the first place. So it's science and building our understanding of the world, whether it's the physical world, the living world, or the human world, that allows us to create new technologies. What gives science its transformative impact is that by understanding the world, we can change the world, and only by understanding it. Now, this is not something that we've discovered in the recent past. In preparing this talk with a wonderful policy analyst at the ERC, Ben Turner, Ben came up with a number of illuminating quotes from 4 centuries that I will use to illustrate this before getting back to what we can, want, or should do today. Scientists have seen from the beginning the importance of understanding how the world functions. So Bacon argued in 1620 that, I quote, and it's a wonderful quote, "Nature can only be commanded by obeying her." He didn't even say understanding, but obeying her. We have to use what nature gives us to control nature. He went on to say that by pursuing science, humanity would be raised above the conditions of helplessness, poverty, and misery, while coming into condition of peace, prosperity, and security. So he saw the use of understanding the world. The very positive and optimistic view, of course. A couple of hundred years later, the Marquis de Condorcet gave further expression to this optimism in his sketch for a historical picture of the progress of the human mind. He predicted unlimited progress, not only in science technology, but in morality and society. He wrote about the equality of the races and the sexes and peace between nations more than 200 years ago. Again, huge optimism, and we wish it had come out all that way. Albert Russell Wallace, a contemporary of Darwin's, who also worked with Darwin on evolution, called his book about his century The Wonderful Century. Again, this optimism and this appreciation for what science could do for humanity. Of course, this naive optimism and confidence was shattered a bit by the events of the last century, actually already the 19th century, but the wars in the last century were, of course, a death knell to this childlike optimism, where it turned out that technical and material progress did not automatically lead to moral progress or an end to wars. Instead, science even produced the terrible new weapons that did so much damage. So that's why in the last century, scientists began to be more circumspect, and I want to quote Enrico Fermi when he talked about science in the 20th century. He said, "Some of you may ask, what is the good of working so hard merely to collect a few facts which will bring no pleasure except to a few long-haired professors? Because only a few specialists at best will be able to understand them." In answer to such questions, I may venture a fairly safe prediction. History of science and technology has consistently taught us that scientific advances in basic understanding have sooner or later led to technical and industrial applications that have revolutionized our way of life. He warned, what is less certain, and what we all fervently hope, is that man will soon grow sufficiently adult to make good use of the powers acquired over nature. Again, I don't think we've seen this growing up fully, but we must retain hope. But here we are at the start of the 21st century, faced with the realization that our own actions have been causing irreparable damage to nature and to warming of the atmosphere, with dire repercussions for our planet and our way of life. Given this, it is perhaps not so surprising that some people turn against science and the idea of progress and the goal of economic growth. But let's not forget that there has been progress and that many of the dreams of the Enlightenment have come to pass. Many of us live in unprecedented levels of material comfort and health compared to our ancestors. And again, we mustn't forget this when we think about research and intellectual activity as a cultural activity that we can afford. Europe can afford to do this. And this is not just, this progress is not just a rich world phenomenon. In 1800, 43% of the world's newborns died before their 5th birthday. It was perfectly normal to lose every other child. In 1960, child mortality was around 20%. Over the last decades, it's gone down to 4.3%. In rich countries, sometimes as low as 1%. And note that that percentage does not correlate with wealth alone. So there's also a role of politicians for using what science does. Some of our richest countries are not as low as 1%. So maybe Bacon and Condorcet have been at least partly proven right in their optimism and justify us retaining our optimism. At the same time, while we acknowledge this progress, we have to admit that we still face tremendous challenges. We can acknowledge that many of the problems we face are of our own making, and at the same time, trust that we can solve them. Let's remember the original goal of science, namely to understand the world. In the context of what to do with scientific findings and that understanding, let's quote Richard Feynman. Scientific knowledge enables us to do all kinds of things and to make all kinds of things. Scientific knowledge is an enabling power. Knowledge is an enabling power to do either good or bad, but it does not carry instructions on how to use it. That has to come from elsewhere. So as scientists, we must understand this power that our findings have, but we must also accept our responsibility for the ways in which our findings are used. Creating a culture of ethics and integrity in research is as important as fostering a spirit of inquiry. That's what I've really tried to push in my former role as EMBO director. The ERC has tried to push. They're really exemplary, and we all must continue to push together. Of course, we can and must be part of the solution of the problems we face now, and we are. Remember, it was the scientists who first detected global warming. And science must play its part in trying to reach a new equilibrium. We must actively undo what we've done. I do not doubt that new and improved technologies can make major contributions to addressing some of the many challenges we face. But from the social sciences and humanities, we can also contribute new ideas, new insights, new theories, and new algorithms about how to live sustainably, how to confront inequality, how we might work and live and educate ourselves together in future. So there's also a potential problem with this thought of freedom of inquiry. Since the earliest days, scientists have fought for freedom and inquiry. Among current examples of institutions that really promote this are, for example, here in Germany, the Max Planck Society, which operates on the so-called Harnack principle, which is named after Alfred von Harnack, the first president of the predecessor of the Max Planck Society, the Kaiser Wilhelm Society. He advocated the right of researchers to work independently of government or private requirements, and unencumbered by bureaucracy, thankfully. And to this day, that's what Max Planck does. It's built around individual researchers who themselves define their research subjects are given the best working condition and freedom. In the UK, in 1918, Richard Burden Haldane from the big spread and famous Haldane Firm chaired a committee which produced a report recommending that general research should be under the control of autonomous research councils. Those should be free from political and administrative pressures that might discourage research in certain areas. And in the US, a 1945 report to the President of the United States called for an expansion of government support for science and the creation of the National Science Foundation. Vannevar Bush, in his piece Science, the Endless Frontiers, stated, "Scientific progress on a broad front results from the free play of free intellects working on subjects of their own choice, in a manner dictated by their curiosity for exploration of the unknown. Freedom of inquiry must be preserved under any plan for government support of science." What a statement. Maybe that's why the US is doing so well in research. Now, the ERC operates according to similar principles. It supports excellent scientists from anywhere in the world, of any age, and from any field in research, including the social sciences and humanities. There are no predetermined targets or quotas. It provides substantial long-term funding for up to 5 years. The only conditions are that ERC-funded research must be based in Europe and be adventurous and take risks. This approach can be confusing to politicians, policymakers, and administrators. They're under pressure to deliver results in the short term, all the more so when we are confronted with major crises like now. It's very promising it's very tempting to promise to fund scientists to solve climate change. It's much harder to say, we will fund scientists to develop new knowledge. Scientists understand this political pressure for immediate results, and that there are indeed many pressing problems in the world. They share these views. So why do funders such as the ERC not set priorities and instruct our grantees to solve specific problems? This is certainly not because we care more, or other organizations care more about the needs of scientists than the needs of society. In fact, like I said, researchers share the concern and the desire for solutions. But the philosophy of the ERC rests on the idea that researchers know best the most promising research areas to explore. I'll give you an example of that. I chaired the ERC Synergy Grant panel a couple of years ago, or 3 years ago. Wonderful grants, they are multidisciplinary, and our panel was covered the whole breadth of the life sciences, and was composed with people who represented the life sciences. It turned out that the grants that had come in, the applications that had come in, there were an exceptional number on the environment, population biology, interaction of beings with their environment, evolution, etc., etc. Mind-boggling, and we did not have anywhere near expertise on the committee for this onslaught of grants in a new field, because this was new. And it just turned out that the best grants by the best scientists were on pressing problems, or on establishing the basis, the basic knowledge of problems for problems that we would need to find solutions. So we don't need to tell them, they vote with their feet. So putting all our resources into so one of my major aims as a new president of the ERC will be to fight for sufficient funding for frontier research. If we look at the journey which got us the MRC vaccines, that's another example. We need to look no further. It was independent, basic research that was immediately deployed with amazing speed to find vaccines. This is not unusual. Any technology or project or service that we use at the moment is a combination of many components. Each of these has its own story, often going way into the distant future. The idea of combination of technologies and of findings may sound trivial or obvious, but it isn't. It's important to understand, because with each new understanding we acquire, we now have the possibility of combining it with all other knowledge we already had. This is called combinatorial explosion. And again, this is what's promoted by allowing scientists to do what they think is most important, and just providing elements that others, if they so wish, can use to turn into application. Once we understand the combinatorial nature of technology, and we can see why science, innovation, and technological progress are not linear. Each feeds on the other in a constant series of feedback loops. There is no point at which the science stops and the so-called innovation begins. Problem is when policymakers start to believe that they can simply order researchers to come up with whatever they think is necessary. Then we end up with absurdities such as researchers being asked when they ask for funding, please tell us what discovery you are going to make. A contradiction in terms. If you know what you're going to discover, it can't be a discovery. And which societal challenge you will solve. So I've argued that the transformative event of science comes from discovering and understanding, and thereby being manipulated in even greater detail natural phenomena. Combining these understandings together in ever new and unpredictable ways is another major way forward. It's been clear that researchers must have freedom to explore these phenomena. That is why funding cannot be short-sighted. To maintain a healthy research system, it is right to invest in long-term, curiosity-driven research. Some consider this approach to be idealistic, and it is idealistic. Of course it is idealistic, but it is also necessary in order for science to have its maximal impact for the benefit of society. And as long as I'm president of the ERC, I will continue to make this argument and do it as long as it takes to be heard. Thank you.
Thank you so much, Maria Leptin. If you would like to stay with me for a minute here, there might be some questions. We have like 5 minutes or so if you would like to take some questions from the audience. If there are any, I have a question anyways, but I would like to give you the opportunity. Professor Ziegler, there should be a microphone somewhere
I
here.
give it away.
Oh, there's a microphone. There's the microphone.
Perhaps it's too early to ask, but the question will be, of course, whether you feel that the European Commission and so have understood or value the value of basic research, which in the end drives things. Perhaps you'll just watch that now.
You've, of course, hit the nail on the head. It will depend on them what the next framework program will allow us to do and how much money they will allocate. Many politicians are very enlightened and are happy to see this. Of course, not everybody. I think, but politicians listen to their constituencies. Their constituencies are their voters. The voters are the citizens in the countries. And I actually believe that people have an inherent appreciation of curiosity. People care about black holes, even though they do them no good. People care about how the Lascaux caves were painted, what paint was used. They care about it. They read Kahneman, Thinking Slow, Thinking Fast. That's he developed this while walking around campus, paid a nice salary, but they care. So I think it can be made clear to everyone that the value of base, and I'm deliberately quoting those things that don't have an immediate impact. I think there is room to convince the citizens in Europe to remind them of the value of basic and curiosity-driven research. And that's the way. It can't be done overnight. New methods for reaching them will have to be found. But we're exploring that.
Let me just follow up on that. Who is in charge, or who has to convince voters in Europe that basic science is just as important as you say? The researchers themselves, the research organizations, the politicians, who is in charge of actually building up this trust?
Everybody, everybody you named. And it's very nice the ERC grantees are actually very good in helping and engaging with, and Jean-Pierre has found ways of communicating science. So I think everybody. Many research organizations actually force their grantees to do public outreach, which I think is good. And some scientists are very good at it, but some are not very good at it. Some are very good at sitting in their darkened room and thinking, or
so should they be forced too?
I think there's no point. I think there are people who are good communicators. You probably are one. So there are people who are journalists, bloggers, movie makers. Everybody listens to Attenborough. Now, there must be people out there, and I know there are people out there, and I think engaging with them. It also has to be done in the national language, so there's no point in somebody sitting in Brussels and lecturing. So I think it requires a long-term approach, hopefully helped by our grantees, but also helped by engaged journalists, and I know they're there.
Would you agree that the overall trust into science in Europe is pretty high still?
I think it varies a lot. I mean, and it's not, come on, you know this. I guess we're not quite as bad as the US in being seduced by fake news. I think education in many places is good and trains people to critical thinking. But, you know, you have to be on the lookout all the time. Ultra important to give children the basic understanding of what science does and how it works.
Thank you.
Never enough.
Please, go ahead.
Wonderful,
Martin,
Dr.
thank you. Hi.
Maria, as usual. I have a general question. So science and education are two things which never can be fully separated and probably shouldn't be. But sometimes in Europe, those scientists who educate most of the students, most of the future scientists, are not living in institutions that necessarily provide the best framework for doing science. So what's your view on this, and what's the ERC able to do to improve that situation?
I think the ERC can't do anything. It's a structural problem, and again, it's national. You know, the US Howard Hughes system, which is equally generous, and as the ERC, funds researchers within universities. The Max Planck Society funds researchers outside universities. In some places, I mean, I know Germany well, I am German, you know, there's good connections, but not everywhere. I agree with you. The top scientists should be in close to training institutions. But really, I think the most important step is actually at school.
Thank you. We have time for one more question, if there is one. All right. Then we'll just move on to the presentation. Thank you so much, Maria Leptin. So ERC is about funding science. So basically, it's about real science, and now it's time to hear about some real science projects, about some research that has been done or is being done by researchers or grantees, ERC grantees. Let's learn about 4 concrete examples of excellent research, all from Berlin, 4 ERC grantees, 1 from each BUA partner university. What is the transformative effect to their science? We will find out now. And it's also a special challenge for the researchers. Their research is complex, but they only have 7 minutes each to explain it. And they must be very focused because they're only allowed to use a few slides without much text. So not the way they would normally present their research, but they all took on the challenge, and I'm really excited to present them to you. And we will just get started with Matthias Rillig. And just for all those who present, those 7 minutes, you know, I will stand up after 5 minutes, so then you know you have 2 minutes left. And let me introduce Matthias Rillig to you. He's professor of ecology at the Freie Universität Berlin. His team addresses a range of questions related to global change, biodiversity, and terrestrial ecosystems, especially soil. He received an ERC advance grant for his project Gradual Change. Today he will be talking about multiple factors. Professor Matthias Rillig, everybody.
Yeah, thanks a lot. Good evening. I'm pretty excited to be talking to you today about this topic has just been mentioned. And we currently hear a lot about climate change. I mean, currently the COP26 conference is going on, and you hear it in the news all the time. And I have this presentation, but I also have one mission. And that mission is that I want you to think more about global change rather than climate change. This is the one thing I would like to take you home, that you take home with you. And so here's why. So of course, climate change is a super important topic, but we do a lot more to this planet than just change the climate. We have invasive species, we have elevated atmospheric CO2 concentrations, we have atmospheric nitrogen deposition, we have microplastic, all kinds of chemical pollution, the list goes on. And this is also important to take into account. Now, what we did to start is we looked at the literature, we looked at a couple thousand papers, and we screened for how many factors are people actually examining in experiments in terms of global change and soil and terrestrial ecosystems. I don't know if you can guess how many factors people generally examined in their papers. 98.2% of all papers ever published on global change did 1 or 2. So only a super tiny proportion of all papers by scientists ever published from experimental work looked at more than 2 factors. That's pretty crazy because we know that soils and terrestrial ecosystems are going to be affected by a whole range of factors. Think, for example, about urban soils here in Berlin or agricultural systems. So you might also ask, why is that so? Why do people not study more than 2 factors? Well, the reason is actually the combinatorial explosion problem that you mentioned. That is like when you sit around a table, looking at a mathematician right there, this is like the number of clinks between the glasses of people sitting around the table. It goes up very, very rapidly with the number of people sitting around the table. So if you're looking at 10 factors, 10 factors, drivers of global change, you have 2 to the power 10 combinations if you only look at 2 levels of each factor. That's 1,024 different treatment combinations, and nobody can do an experiment like this. So what did we do? We did a little trick. We asked a slightly different question, but I did an experiment with 10 factors that's shown here. And what I did is I had a pool of 10 factors, and I sampled randomly from that pool of 10 factors a given number of factors along a gradient of increasing number of factors going from 0, the control, to 1, 2, 5, 8, and all 10. So in factor level 2, for example, I randomly pulled 2 global change factors from my pool, maybe microplastic and warming. This was replicate number 1. Second replicate was maybe salinity and herbicide. Second replicate. So all of the replicates of the factor level 2 had different combinations of factors. This is actually a pretty crazy experiment. It's never been done. You're literally comparing apples and oranges, and my expectation was what would come out is chaos. But what came out, the result was not chaos at all. It was super surprising. What we found, and I'm told show no graphs of data and whatever, so I'm going to show you that, you're going to have to believe me, but it was a very straight line, an unusually straight line for an ecological study with so many factors active, that decreased with the number of factors that we had switched on in our experiment. So the more factors we had switched on, the more biodiversity we lost in these soils, and the lower were the soil processes and soil-borne ecosystem functions. Now, this was really weird because the single factors that we had applied had either positive effects, negative effects, or neutral effects. But when you combine them, what came out was a just steady decrease in biodiversity and ecosystem functions and process rates. And how can you explain that? The answer, we don't know. Actually, we have no idea what's going on there, but I'm going to try to explain it to you like I would explain it to my mom. So you have a job in this ecosystem. Your job is to decompose organic matter if you live in the soil as a fungus or a bacterium, let's say. This is your job. So transfer the situation to you. You have a job. You have to write a report. That report has a deadline. Okay, so now 3 things happen. I'm going to only go 3, not 10, but you can complete it in your mind. So the one thing that happens is your super fun neighbor shows up at your door with a bottle of red wine, and he wants to celebrate. Now, this is very positive because you want to celebrate with that neighbor, but you also have a report to write with a deadline. And so then a neutral thing happens. Your dad asks you a complicated tax question. But this is neutral. You have to answer to that question. It doesn't take much effort, but you still have to do it. It also takes effort away from doing your task, writing the report. And then, of course, whenever you rely on the internet, it cuts out at home, so you have an internet outage. And that is a negative effect. Now, these things all happen to you at the same time, so it doesn't really matter if it's positive or negative or neutral. The end result is this adds to your stress. Now imagine this would be happening in the soil. Now imagine also this happens in the soil. You're not just one person. You are one species of many species. In our tiny little soil samples, we had thousands of species of bacteria, and we had hundreds of species of fungi, and God knows how many protists. We measured them all. So this is slightly more complicated than what I have depicted here. Now this is in the end, kind of bad news. Sorry, because with more factors, and we have all these factors, this is not fantasy, this is what's really happening, we will be losing biodiversity in soil ecosystem functions and process rates. But there is also a silver lining. I think this is very important to emphasize, because it means that if we can switch off some of these factors that are inevitably acting, we will be moving back along this line and maybe towards more biodiversity and better ecosystem process rates. But what's important is that we move the conversation, and this is how I finish up, to global change. We cannot really afford to just talk about climate change. And we also need to do more research. And yesterday we've started this experiment here. This is the first global change experiment that I know with 10 factors. So you can see open top chambers here for passive warming, and rainout shelters are for drought, and the other 8 treatments are soil-borne treatments. So this is how we hope to find out more about these many, many factors. This is actually on the Humboldt University property, even though I am at FU. Bua. Thanks very much.
All right. Thank you so much. Again, there is the opportunity for some questions, and I'll take the opportunity to ask you the first question. If most research has only looked at one factor or 2 factor, and the results of your experiment, what does those results, what do they mean to all this research that has been done? Does your experiment render them in a way senseless? Because they have not really looked at the whole story?
Not really, because these single factor experiments are of course also important to understand mechanism. And we need to understand mechanism if we want to predict. Now, the downside, so to speak, because you never solve everything with one experiment, of this approach is that we don't really know which factors and which combination of factors are really responsible for the effects. We used all kinds of statistical models to tease that out, but we cannot really determine that because there's too many factors that are acting. So no, we still need also the single factor experiments and the combinations of maybe 1 or 2, because this is the way we find out about mechanisms of how these factors act. And actually what I did is, based on all these single factor studies, I went ahead and did a classification, like a phylogenetic tree of all these global change factors, like really a tree of similarity of all the global change factors based on traits. And we could use that based on basically the single factor research to predict a little bit of what came out of this experiment.
Thank you so much. Any questions from the audience? Günther Ziegler?
I guess we are trained to sort of sort our things into most important things first. Like, I mean, out of the factors, what's the most important one? Deal with that one first. And the second question would be, things are related, of course. Climate change in the end is not a factor, something that things start with, but it's something that's caused by other things like CO2. Does it make sense to sort things that way?
Yeah, no, I think it does. I mean, it doesn't preclude the fact that they're also related, like what you had explained, for example. This is also correct. And I get this response a lot, you know. Haven't we really covered our bases by looking at warming and drought and elevated CO2 and nitrogen deposition from the atmosphere? Those are the big 4. Almost all papers are on these big 4. And the answer is, I don't think so. I think we need also these other factors because they are real, they really occur. Like our lab works a lot on microplastic, and actually in our sea grant. And microplastic is also everywhere. It literally rains down from the atmosphere, even the most remote regions on this planet, because it comes off your clothes. It's literally everywhere. And so this argument can be made for many other factors. So I think we are missing some very, very important results by just looking at the big 4, as they are called. I think we need to extend that set of factors.
Thank you so much. We'll take in those 2 questions, Professor Bourguignon, and then you, and we'll take them together, and then we'll get back to you. Please, go ahead.
Thank you. You didn't mention at all one thing which I'm sure you're very, very much aware of, which is the value of thresholds. That is, there is a plasticity, which means that for a certain range, basically, even if there is a difference, it does make a difference. But then when you pass a certain threshold, then all of a sudden things can change drastically. Can you also have a look at this with your experiment?
We did.
Maybe we just take the second question in, and then you will answer.
Thank you. All very plausible and all very scary at the same time. So I have another question to evolution. So microbial systems are known to be extremely versatile in adaptivity and in evolution. So if you add combinatorial stresses on them, do you expect an acceleration of evolutionary trajectories that we haven't seen before?
Please, go ahead.
So to the first question, we did look for tipping point behavior in our data set, and we found some evidence that there may be tipping points going on, but it was actually not a break point in our data, but it was really a continuous decline, which was really surprising. But you're mentioning another point, that it is actually we only looked at 2 levels of each factor. And the levels were actually chosen specifically to be very conservative. So we didn't add like a lot of this material, or we didn't increase temperature by a huge amount, or we didn't have a very severe drought. And we tried to have these levels informed by actually measured values that would be present in Germany, for example, maybe going a little higher to simulate a hotspot. So it was a pretty conservative approach. But what we are missing actually is really these response surfaces. Now, as you can imagine, if you have so many factor combinations, this was a pretty difficult experiment to do. If you also have different levels of each factor, it gets very quickly out of hand. But we are working on that a little bit. We have some ideas on how we can actually combine these more response surface approaches with many, many levels for one factor at least, and then maybe the other factors get switched on en bloc or off. So this is the way I think we're going to do this in the next experiments. But we had that definitely in, we were thinking about this all along, because this is an obvious point, because we only have 2 levels of each factor. About evolution, we have some people in the lab. Actually, this was one of the things in the ERC grant to work on evolution, but this part with the multiple factors we haven't addressed yet. I am quite sure it will change evolutionary trajectories, and I'm quite sure that even after the 6 weeks of this little short-term incubation, we will have had evolutionary changes in those microbes, no doubt. But how to predict those and what are the consequences of these evolutionary changes for the outcomes that we see, that I don't know. Yeah.
Matthias Rillig, thank you so much. And you can just see he's really excited. He's still excited, and he has fun, doing the research he is doing. So thanks so much. And we're moving on to the Humboldt-Universität. Susanne Schreiber is a professor at the Institute for Theoretical Biology at the Humboldt University and employs mathematics to understand how our brain works. As theoretical neuroscientist, she also chairs the Bernstein Network of Computational Neuroscience and is vice chair of the German Ethics Council. And she received the ERC Consolidator Grant. Title of her presentation, How the Personality of Nerve Cells Shapes the Rhythms in Our Brain. Please, go ahead. Thanks so much.
Thank you very much. It's a pleasure to be here tonight. I'm particularly grateful to the ERC funding, so it's very great to see the new president here. And I'm going to tell you a little bit about this project, which is about the brain. And what you see here to start with is a network of symbolized cells. And what we know is that the connections between the cells are the really important thing. So when we learn, then these connections change. That's actually quite intuitive. So the question is about who talks to whom, and if you think about a scenario like that, then you'll see or understand that it makes a difference. If you look at different situations here, the outcome in the end, if these people have to solve a problem, may be different from the first example to the second example. That's what all neuroscientists take for granted. Something that is not so well taken into account often, but is the creator of my lab, is that there are personalities. So if you think of this example here, then you see, even though you may have this constellation here, then if these people are very accessible and very empathetic, you may get a different result if they have to solve a problem compared to if they were all choleric and uncaring. So neural personality, that's the same for the neurons, matters a lot, not just who talks to whom. So if we think about nerve cell communication, what does that actually mean? So how do neurons talk? What they do is they elicit these pulses that you see here of electrical activity. They pass them on to the other cells, and the other cells listen. And what we know is that we have 3 personality types of cells. I'm going to put that a bit simpler here. We have the blue cells, which I call the slow starters. You have the jump starters and the it depends starters. The slow starters start with pulses once they are triggered that follow, or that are not so many, and then you have to excite them more to get more pulses. The jump starters, if they're triggered, they jump right away to a high pulse rate, and the it depends starters are a mixture between the two. Now, this is not what they're termed in literature. Actually, we have known 2 of these types since the 1950s. So that's the classical type 1 and type 2 described by Hodgkin. And this one is called the homoclinic, and it was kind of rediscovered by us in the sense that everybody believed they're not there in the brain, but they have special properties, and you can find them mathematically, you can predict they must be there. And I'm going to tell you why these properties are so special, because that's the core of the grant. So, you may not be surprised to hear that for communication, it's not only about talking, but a lot is about listening. And that's the same for nerve cells. So if you look at the type 2 here, then this is a kind of self-absorbed guy, because if he elicits a pulse, which you see here with the star, then it first needs to recover from his own speech before it can listen to others, because what this curve here shows is when does it listen best to the other cells. And you see there's a temporal delay here. Whereas the homoclinic one, he's very special, because what happens is if he talks, if he elicits a pulse, then he can right away listen to the other cells. And that gives a very nice property if you join these cells in the network. So think of 10 neurons coupled in a network, here in an all-to-all fashion. And then what you see here is the pulses that these 10 neurons elicit. Here they are kind of disordered. If you now take the same network, and those are now these homoclinic personalities of cells, then you see that they all fire in concert. So we have a synchronized activity, and what this means is if you look at the level of the EEG signal, so the summed signal of activity in the brain, or the local summed activity, then if you sum these up here, you won't see a brain rhythm, or only a very weak one. Whereas here, if they're synchronized, you'll see these large elevations, these large oscillations. So these guys, these homoclinic cells with this personality, they can give yield, or they can give very strong brain rhythms. So one last thing I need to tell you for you to see what is the nice thing about the grant is nerve cells can actually switch their personality. So it's not fixed, but they can turn from a type 1 neuron, for example, to a homoclinic neuron. One thing that we found is that a temperature increase suffices to do that. So when you increase temperature, then normally a type 1 will become a homoclinic neuron. What follows from this is also the brain rhythm is changing. So you see here a very weak brain rhythm. If you then increase the temperature actually only by 2 degrees Celsius, you start to see that you have these really large elevations, so you get a strong brain rhythm in the EEG signal. And this is what this grant is about. So we want to understand how personality switches can give rise to very strong brain rhythms, because this may be interesting for pathologies like epilepsy. It may be one mechanism how epilepsy can be triggered in its initial phase. So the thing we're going to look at is the trigger factors. I already mentioned temperature here. Another one is really nice, and that is if the brain does not have enough energy. So if the neurons get exhausted, then the neurons also turn into homoclinic spikers, and this is something that we often see in disease. So we often see that either temperature is increased that can trigger an epileptic seizure, or that energy goes down, and there's many more factors. So just to summarize, we're going to look in this project at the mechanisms that can trigger these brain rhythms based on neuronal personalities, at the trigger factors, and we hope to find that the brain is full of homoclinic personalities, which we don't know yet. What we use to achieve that, or to discover that, is actually mathematics. You see that on the left-hand side. So I'm not an experimental neuroscientist. I'm using mathematics, mathematical simulations to understand these things and to come up with predictions. Yeah, and I'm very happy to take your questions. I thank you for listening.
Thank you, Susanne Schreiber. Are there any questions from the audience? Can hardly see you, but I can kind of guess if somebody is. All right, otherwise I would like to start. You said, could we go back to the last slide maybe? Is that possible? Last question, is the brain full of, is it full of homoclinic personalities?
This one? Or
Yeah, this one. The last question. I don't really get this. I get the first two, but why is that important? I thought that they can change anyways. They can be either this or that. So how is that question relevant if any cell can be a homoclinic personality at any given time?
The difference is that this is what we find in theory. This is what we find with mathematics, where we can actually really prove that if you start out with one cell type and you change these factors, you are bound to end up with the other cell type. But whether that really is what nature does, because so far since the 50s, people have only found the type 1 and type 2 neurons, only have discussed these, and it's very difficult to distinguish, is there this homoclinic spiker in there or not, because he often looks like either type 1 or type 2. And these listening curves, they're very hard to measure. So essentially, we don't know in the real brain whether they are there. We predict from our work they should be
just
there.
in the model
They should
there.
be everywhere, exactly, but we're going to find out whether, in collaboration with experimentalists, whether that's the case or not.
All right, thank you. Yeah, please, go ahead.
Sorry, you just mentioned the word experimentalists. Can you tell us something about what kind of experimental data you're comparing your work to?
So we're very fortunate to have several collaborators here in Berlin. What we start with is slice work. So where you take the brain, you slice it, and then it's an in vitro approach, because there we can control the cells in isolation. So we can cut synaptic pharmacologically, rule out the synaptic connections, and then can start to manipulate. What we hope to go for is in the end also an in vivo approach, where we can use in vivo data and use machine learning techniques because that's difficult, because we have also the network input, but we hope to learn how to classify these neuronal personalities even if the network is intact, to distinguish them.
Yeah, one question by Günther Ziegler.
We learned that there's only 3 types of cells. They're type 1, type 2, and homoclinic. How do you know? Are you sure? Or is this just a simple model?
It's not just a simple model, but it's a simplification, a pretty drastic one for the purpose of this talk. So this is true for the regularly firing cells, that fire in a nice rhythm. We also have other types of cells that have bursting patterns and so on, and they can be classified into different classes. So I'm not talking about those. But the 3 classes are, as you're a mathematician, I'm allowed to answer this question. It depends on the mathematical bifurcation that underlies the initiation of the limit cycle, so of this onset of repetitive spiking. And there's only 3 types that yield a roughly constant amplitude. There's a 4th type, but then it starts out with tiny spikes, and that's what I'm not looking at. So 3 mathematical types.
All right. Thank you so much, Susanne Schreiber. So let's move on to the technical university. Begüm Demir is the founding head of the Remote Sensing Image Analysis Group at the Faculty of Electrical Engineering and Computer Science of the TU Berlin, and she also leads the Big Data Analytics for Earth Observation Research Group at the Berlin Institute for the Foundations of Learning in Data. You perform research in the field of processing and analysis of large-scale Earth observation data acquired by airborne and satellite-borne systems. You received an ERC starting grant, and you will now tell us about your research, accurate and scalable processing of big data in Earth observation. Professor Begüm Demir, please go ahead.
Thank you very much for the nice introduction. It's a great pleasure for me to be here to present our initial research discoveries on my Big Earth project. So, it is all on accurate and scalable processing of big data in Earth observation. And at our team, we especially do foundational research in terms of big data management and machine learning for Earth observation. So it's very important because with the advances in satellite technology, remote sensing data archives are growing at exceptionally fast rate, not only in volume, but also in diversity. As an example, with the Copernicus program, that is the European flagship satellite initiative for Earth observation, Sentinel satellites acquire almost 12 terabytes of data per day, and at the moment, the Copernicus data archives has the size of almost 20 petabytes. So the large-scale Earth observation data is the great source of information for several different Earth observation applications. And the information that we can discover by analyzing satellite images can be very helpful for several different applications to monitor the health of our planet. Let me give you an example. So here you see a Sentinel-2 image that is a multispectral image acquired after a very big forest fire on the southern part of France. So the brown area here that you see is representing the burned forest areas. As you know, analyzing burned forest areas is helpful for climate change and ecological studies. And if we can find all the burned forest areas, for example, given in a geographical location or duration of a specific time or so on, this can significantly contribute to studies on climate change and ecology. So this kind of study can be done by digging these Earth observation data archives, because this information is hidden there, but it is a challenge because we need scalable approaches to handle these petabytes of data, plus we need accurate approaches that can handle the complex content of multimodal heterogeneous Earth observation data archives. And actually, this is the main aim of my Big Earth project, where we are developing search engines that can operate on the semantic content of the satellite data. Here you see the application in terms of forest fire, but we are developing systems that can deal with several different applications, so it's independent from the selected application field. So, the methods and algorithms that we are developing contribute to the foundational research in terms of knowledge discovery for querying and indexing multimodal Earth observation data. We are recently in contact with space agencies in Europe, in particular European Space Agency, to deploy our tools into their systems. Because before Big Earth, the operational Earth observation systems was operating only on the keywords, okay, not on the semantic content of the satellite data. But when we consider large-scale data, it was not very sufficient. Okay? So with Big Earth, we are changing the paradigm in this line. Okay. So, independently from the Earth observation application that we are dealing, one of the most critical issue was to have access to training data. So this is very important if you are dealing with machine learning, deep learning. You need a large amount of data, and it is very difficult if you are dealing with Earth observation applications. So to handle this problem, to drive our knowledge discovery systems for search and query purposes, we developed Big EarthNet. Okay, so Big EarthNet is the large-scale Sentinel archive that includes data coming from 10 European countries. And actually, we are very proud of its success. Since it is first released almost 2 years ago, it has been downloaded 10,000 times, more than 10,000 times by many organizations, including European Space Agency, NASA, German Aerospace Center, and so on. It is publicly available at its website, that is bigearth.net, and also it is available on Google Earth Engine, TensorFlow, and Radiant ML Hub. Okay. So, in order to help to the users to interactively visualize our data set, we recently developed a portal that we call it as Big EarthNet portal. So that portal, thanks to the technologies that it uses, it allows the users to efficiently and effectively discover the Big EarthNet. By just selecting the geographical area, by just selecting polygon around the geographical area of interest, the tool returns the images to the user with specific characteristics. For example, you can see land use, land cover classes present in the area, and also the statistics driven based on this information. Okay. So, in a nutshell, Big Earth is aiming to develop accurate and scalable systems for indexing our Earth to discover in a quick way and an accurate way information about it. And I believe that the systems that we are developing through Big EarthNet will significantly contribute to recent initiative of European Commission, that is Destination Earth. Destination Earth is aiming to develop digital twins of different aspects of our Earth system, and then infrastructures, models, tools to deal with this data. And we are hoping that they will be also taking advantage of our tools that we are developing in the ZRC project. Thank you.
Thank you so much. That was quick. Yeah, please go ahead. First question by Professor Lept.
So your training data set is interesting. I mean, you must give the ground truth of what's in those data. Somebody has to say what the machine is supposed to learn.
Yeah.
So, but you can only give things that you already know about those data. Yeah. So how do you discover new things? Or how does the machine learn to discover new things?
So when we deal with satellite data, if it is a small-scale problem like classification of Berlin, okay, then it is not that challenging, right? But when you do large-scale analysis, it's a challenge to collect this data, and it is almost impossible to do it manually. So that was one of the challenging situations. And then at the time that we started with our grant, European Environment Agency made public a Korean database of 2018 for whole Europe. So they provided land use, land cover class labels, and I think we were lucky that it just came in our first year. And then we use Copernicus data, satellite data of European Commission, and we mapped this with the labels provided by European Environment Agency. So, and then we made also publicly available our models trained on Big EarthNet, and that made a big impact. Yeah. So the labels are provided by European Environment Agency, but of course they were not targeting our project. It was for other purposes, but we took advantage of it.
Mm-hmm. Thank you so much. Any more questions? Günther Ziegler?
I seem to always have questions. So you describe this basically as searching data and finding things in this data. But one of the things that is certainly interesting, like if you take the Copernicus data, is to integrate them over time, to see change and
of course,
to
yes.
discover that.
Yeah.
So that's sort of one question.
Yeah.
And the other one is how is something like that mapped to something like Google Earth, which of course gives you pictures, again, a rather static thing. And I'm just thinking of that because Google Earth apparently was copied from a Berlin project, TerraVision, that was this recent network thing. Right?
So, and these are really very good questions. So, of course, we are very much interested in change detection. And actually, this search engine, we are adapting it for change retrieval. So here, for example, I have shown you a query on burnt forest, but query can be also deforestation, which requires at least 2 images, before and after deforestation, right? And then we can model these pairs of images in the query and search for similar deforested areas. So I think it's a very timely and interesting field in this line, and it is in our agenda. So for the Google Earth, so Google Earth engine took because our dataset is publicly available for research purposes, and Google Earth engine provided our dataset and pre-trained machine learning models under Google Earth engine, and yeah, that's it. But we are we are closely working with European Space Agency. I should admit this, and yeah, we would like to make it European search engine for satellite image search and retrieval. Yeah.
Very exciting. Thank you so much, Begüm Demir.
Thank you very much.
So there's one more partner in the alliance, and it's the Charité. So the next presentation comes from a researcher from the Charité, Suryo Sugata, connecting the brain with machines. This is his research focus. Go ahead. You found out that brain-controlled devices cannot only improve quality of life in paralysis, but may soon transform the treatment of brain disorders such as depression, addiction, or dementia. You also received the ERC starting grant, and we are really looking forward to hearing more about your research. Go ahead, please.
It's very wonderful to be here tonight. It is estimated that more than 1 billion people on this planet suffer from a disorder of the brain. For example, depression, anxiety, stroke, multiple sclerosis, or dementia. But despite all tremendous technological progress, we still don't have the tools to tackle brain disorders effectively and without side effects. So in my ERC-funded project, I am developing the next generation neurotechnologies that will improve quality of life and restore brain functions across various brain disorders. The project builds on the latest advances in brain-computer interfaces. So these brain-computer interfaces translate brain activity into control commands of a digital device. For example, here you see a patient controlling a prosthetic arm to grasp a coffee and drink. Or you can even use these BCIs for controlling several prosthetic devices. However, implantation of these microelectrodes entails the risk of infections and bleedings, and there's no certification to use these devices on a long term. And none of these patients have ever used these technologies outside of the laboratory in their daily life. So in my research, my team and I, we are developing non-invasive brain-computer interfaces that do not require a surgical procedure. Here you see a number of quadriplegic patients who are all in a wheelchair and who are unable to grasp with their own fingers. They are unable to grasp a book, a cup, or a piece of paper. So we have equipped these patients with a neural exoskeleton that would translate their brain activity associated with the intention to close their fingers into actual closing motions. And here you can see that these patients can securely grasp and manipulate these different objects of daily life and restore their hand function. The system could also be used outside of the laboratory. So this gentleman went out to a restaurant and ate for the first time after several years with a fork. We asked the patients what they would like to do with this new ability. And there is another patient who asked for a plate of potato chips because he hasn't eaten these potato chips for many years on his own. So here you can see in his face how this technology can improve quality of life. Now, we have used this technology also in chronic stroke patients who were unable to move their fingers, sometimes for years or even decades. And after 1 month of daily training, we were very surprised to see that some of the patients could move the fingers again. And we were, of course, very interested to find out what happened in their brain. And what we found is that the brain activity that was associated with the intention to move the paralyzed fingers reorganized after 1 month of this daily training, and this reorganization correlated with the clinical improvement. Now, there is no reason to believe that this principle cannot also be used for restoration of other brain functions than movements, for example, memory formation, cognitive control, or emotion regulation. And the state-of-the-art brain-computer interfaces on the conceptual level, they translate brain activity into sensory feedback, and the sensory feedback influences brain activity. And we have found that this closed loop leads to functional and structural reorganization of the brain. Now, the next generation brain-computer interfaces, they will also translate brain activity into sensory feedback. But brain activity associated with brain functions like memory formation, emotion regulation, or cognitive control. We will add adaptive brain stimulation to foster stabilization and improvement of brain function. Now, the big challenge is to identify the neural substrates of these different brain disorders, because we can't simply ask someone to be depressed and then stop it like a voluntary movement. So how can we understand what these neural substrates are? A possible way is to record brain activity in the millisecond to millisecond range and apply brain stimulation. and then observe changes in brain function and behavior. Now, this would allow us to understand the causal link between brain activity and brain function or behavior. But of course, there's a problem. When we record brain activity in the millisecond to millisecond range and we apply brain stimulation, there are huge artifacts that make it impossible to really read out these physiological responses. Now, in our ERC project, we have overcome this problem and achieved a breakthrough in applying electric stimulation, non-invasive electric brain stimulation, while we could restore millisecond to millisecond brain activity in real time. So that's very important, in real time, so we can do this in real time. It's not after processing, but we can do this in real time. And we applied this new technology in, for example, an application where we target frontal theta oscillations that are linked to memory formation, often affected in the elderly or patients with dementia. And when we did this, we could find that indeed millisecond precise targeting of these frontal theta oscillations can improve memory performance in the image or picture recognition task, while open loop simulation or non-specific stimulation did not. Now, a critical point here is that we have to increase spatial resolution of neuroimaging tools. So these frontal theta oscillations, they are very easy to catch, but if it comes to other brain functions, very difficult to understand the neural substrates. So here you see the latest generation of quantum sensors. And these quantum sensors can offer non-invasive assessment of brain activity at unprecedented spatial resolution. And here in Berlin with the Physikalisch-Technische Bundesanstalt, we're implementing these new sensors into the ERC project. And we'll build the first of its kind quantum BCI closed-loop brain stimulation platform. What we also do is we are developing novel brain stimulation techniques based on temporal interference of magnetic fields that can reach deep into the brain tissue, where until now we could only reach with implantation of electrodes. With support of the Berlin Institute of Health, we have filed an international patent on this new technology, and we will implement and validate this stimulation in the next 2 years. Now, of course, if we it's very clear, if we use this technology, there are a number of neuroethical questions that are raised, and we, of course, also address them in the project. Now, with this, I'd like to conclude that I'm convinced that, especially with the support of the ERC, science can exert a profound transformative effect on how we treat brain disorders effectively and without side effects. Thank you.
Thank you so much. The slide about the ethics, you were very brief on that. So tell us about the ethics a little bit.
Yes. So I like very much what you said about technology and how it could be also used in applications that are not favorable for, the future of humankind. And now, if it comes to these technologies, we are also faced with this issue, of course, because if we are, for example, manipulating brain activity in ways that people are not aware of, subtle, then, of course, there are a lot of questions that are coming up. And, for example, Chile has now a new law that protects so-called neuro rights, so that everyone has a right to have a protected free will, for example. And of course, we need to do more research, and we need to anticipate what this kind of technology could be used for in a few years. Because you've probably noticed that a lot of private money is going into this field of brain-computer interfaces. Elon Musk has invested a lot of money into this. But that's the invasive approach. But I think from this invasive approach, we'll learn so much about the brain that also non-invasive tools will be able to do these things that we are doing now in the medical field, where it's very clear that it's an imperative to use this technology to improve quality of life. But obviously, this technology could be also used in other ways. And it's very important to protect privacy. Data security is a very important issue. Cybersecurity, if all this data is read out and stored on some server, we need to make sure that it's really safe.
Mm-hmm. Thank you so much. You have a question? Please go ahead. I mean, there's a microphone. You don't? Okay. Are there any questions? So please go ahead.
Okay, so thank you very much for this really impressive talk. I was wondering, compared to the earlier approaches or the invasive approaches, what is the very thing that enables you to do that non-invasively? So can you say that in a few sentences to the lay audience?
Well, I mean, your work actually shows that there are neuronal pools that are responsive to external stimulation. And when we use, for example, closed-loop stimulation, we can target these neuronal populations non-invasively using magnetic fields, for example, that are temporally interfering at some spot in the brain. And as these are larger neuronal pools that are responsive for certain frequencies, we can target these different functions, for example, motor function or memory. But of course, it will be much more interesting to go even deeper into this and have more focality. And that non-invasively is right now a very huge problem, of course. But with these quantum sensors, for example, I'm pretty positive that we can achieve that with higher spatial resolution and especially higher bandwidth. So we can record neuronal oscillations up to 400 hertz non-invasively. And that's really a unique opportunity.
Mm-hmm. Let me just follow up on that. You only observe what's happening now, or can you explain what's happening when you trigger something?
Well, there's a lot of studies that suggest how physiology links to behavior. But we have heard there are so many factors that in the end you can only have theories about this link, and it's all correlative. So in my research, I'm trying really to make a step into causality. So we are changing brain activity in a specific way, and we know exactly how we change it because we are recording brain activity at the same time and see how the stimulation is changing the brain. And then we observe what the result is. And of course, we use then machine learning algorithms to cluster different brain states and different brain functions and changes in brain function. So that's what we do. But in the end, every brain is different. So if you look into neuroscience, you have the Bereitschaftspotential, you all have these Bereitschaftspotentials, some very basic physiological responses, but all your brains are different. Mm-hmm. Yeah? And so we need to be on a personalized level where we use this kind of technology to understand on the personal, individual level what's going on in your specific brain.
Mm-hmm. Thank you. Any more questions? Günter Ziegler, your question this round.
So again, two questions. One is, what is quantum about these quantum sensors? And the other question is, why do you use human brains? Wouldn't it be much easier to start with, I don't know, bees or mice?
Well, these are very good questions. So the first question is, where is the quantum? So usually when you record neuromagnetic activity, there's a technology called magnetoencephalography. And to do this, because the magnetic fields are so minute, you need superconducting devices that have to be cooled with helium. So that was the technology that was state-of-the-art until 5 years ago. And because they had to be cooled, you had to be 4 to 7 centimeters away from the cortex. So very far away from these fields. And now we have vapor cells filled with rubidium, and there are two lasers who are pumping into the gas, and you can change the spin of these rubidium atoms, and you can read out the B field, the environmental field that is actually affecting the spin of the rubidium atoms. And that is the quantum in all this. And here in Germany, we're especially now focusing on synthetic diamond magnetometers. So right now, these optically pumped magnetometers, that's the name for this rubidium cell technology, is mainly done in the US, but it has been developed actually in Berlin at the PTB, the Physikalisch-Technische Bundesanstalt. So my aim is to really bring this technology to applications here in Berlin, and also have, of course, also companies who are going to invest into this technology, to make it available for humans. And that's the next question. So I have a lifetime of, I hope, maybe 3 or 4 decades, and I understand the complexity of, I'm a psychiatrist of humans, and I see that, if we want to bring this technology to humans, we need to do it now. And you can do mice model, you can all do this, it's all very nice. However, the jump into the real application in humans is always unclear. So my approach would be to make this jump already now, of course, under conditions where we know that we are not doing any harm. Now, if you come to my practice here in the Charité, it's not far away, we are using neuromodulation, for example, in depression. And there are patients who were depressed for many years, they had taken medicine, like drugs, have all these side effects, and it didn't work. So they come to my practice, and we use neuromodulation, and we can take them out of depression. And that is what drives me. So what drives me, what motivates me is to use this technology in the clinical case. I hope I answered your question.
Suyo Sokada, thank you
Yes,
so much.
Thank you.
That was fun, wasn't it? I mean, 4 very fascinating personalities, fascinating research they are doing, and the good thing is you can just continue asking them questions, because we're wrapping up this official event now, but there will be a reception following in a minute, and they will stay around, so you can just go up to them, you can just talk to them, ask them more questions, and I think Günter Ziegler has some more questions, so they're happy to answer them. There will be something to drink. Just stay around, celebrate the science, celebrate the BUA, celebrate the ERC, stay around here, and it's in the dinosaurs hall. That's what you wanted me, yeah? Okay, so we'll go there together, so you don't get lost in this great museum, and you don't have to spend the night in the museum. It's in the Dinosaurs Hall, and we just leave there together. Thank you so much again, our 4 researchers, for being here, for providing us with some insight into your science. Thank you so much also to Maria Leptin, to Sabine Kunst, and please stay around, be here for another hour or so. Thank you so much for being here. Have a great evening.